Hello, Dino. Thanks for coming. Nice to see you. I do research on disorders of consciousness. For those of you who might not be familiar with disorders of consciousness, typically they used to be known as in vegetative state. What vegetative state means is that their level of consciousness is so low that they do not seem to respond to the world, nor do they have an awareness of self. Up until about 15 years ago, these people were neglected. They were often put into hospice care, and they were just allowed to die. One of the problems has chronically been that the rate of misdiagnosis for disorders of consciousness patients is in excess of 40%. Now, one of the things that we're trying to do is to identify better ways using fMRI, using FDG PET, to identify through cerebral blood flow, through the neurotransmitter GABA activity, and also through brain metabolism, so glucose uptake, in order to give the clinician a better tool whereby they can render a diagnosis. In Taiwan, where GIMBC is located, usually after about 30 days, patients leave the hospital. And the problem has chronically been that the assessments when they leave the hospital after 30 days are behavioral. And the behavioral assessments are terribly, notoriously unreliable. And that's why we're looking at brain metabolism, we're looking at cerebral blood flow, we're looking at the activity of the GABAergic activity, in particular in the posterior cingulate cortex. And we're trying to use these tools as a better way of rendering a diagnosis and a prognosis. In addition to that, we're running a clinical trial. And for the clinical trial, we're using the drug Ambien. Because paradoxically, Ambien in some cases seems to help patients not go to sleep, but actually to recover, to reboot their level of consciousness.
Hi, welcome. I'm Neil. So the research I do is investigating how the brain organises its overall activity across time. So this is interesting to me because our brain has to change the way that the activity within it is organised according to a constantly changing environment and also according to a constantly changing set of bodily needs. So for example, my brain needs to be processing information in a different way if I'm hungry and I'm out in the street trying to find a snack, as compared to when I'm sitting in a lecture theatre, perhaps paying attention to the speaker and trying to remember and understand the information that they're giving me. So part of the work that I do and related to this is looking at psychiatric and neurological disorders, where I suspect this ability to be flexible in the brain's organisation is altered, producing the deficits that we see in these conditions. So the research I do is mostly in humans, and I use a range of neuroimaging techniques, doing MRI and MEG. And in particular, I'm interested in combining these with ways of measuring neurotransmitters so that we can understand how these neurotransmitters organise our brain's activity.
Hi, I'm Su Xu, one of the faculty in GIMBC TMU. I'm interested in self process in our brain. So self is a very distinctive component that we all have our own self. And I'm most curious how self interact with our rumination. Rumination is ability that we keep thinking for a certain type of thing for repetitively. So in the past experiment or study, it has been shown that rumination, which the processing of the rumination overlap with the self brain circuitry. So it also related to the depression symptoms. So in my current study, I already shown that in the depression, if they have higher rumination, actually they have more self brain-related processing ongoing in those particular group. So in the following, I'm also curious more that in the normal people, how people process self-related or non-self-related stimuli, how does that change their brain activity? At the same time, we also use different type of neuroimaging technique to study our brain. So currently I use EEG, which is high time temporal resolution to record our brain neural activity. At the same time, I also use TMS, which is non-invasive brain stimulation, to find the causality between our brain and its own function. So that's about my study.
Hey, 안녕하세요. My name is Jihan. As you can see, I'm at GIMBC. In GIMBC, we are doing cognitive neuroscience as well as consciousness studies. But I'm more of a biologist. I'm doing biology. Why I'm doing biology? Well, I'm doing biology of time. How the time is processed in the brain. Why time is important. Consciousness. Good question. So, what makes us special? What makes the human being special? Because we think. What kind of thinking? Abstraction is what makes us special. What's the most famous case of abstraction? It's geometry. We have this geometrical concepts. 2 dots on a line. That's already very abstract. And that is probably the most abstract thing that we have been doing in our whole lifetime of humanities. But the really mysterious abstraction that we have is time. Geometry is about space. Time is an abstraction that only we can do it, and it's so special. Why? Because for space, we can, concretely put some dots and connect lines. But time, we cannot do that. Time is always so subject. When you say, let's say when, then you have to involve you. When I spend time with you, for example, we get connected to time. But we cannot have this connection when you are away, when you are not here. You might be in certain time zone, certain day. You're not sharing time. And the meaning of time is completely different. It's difficult to explain. Time is always subject. And then time is also biological. Because we are the biological machinery. And biology is scientific, while the psychology of time is not. So the most scientific way to approach time is to use biology. And I'm using what we call the circadian biology to study how time points are set in the biological question. That's what I do.
Hello, it's Paola. Hi, it's Matteo.
Benvenuti nel nostro laboratorio. Welcome to our lab.
The rest of our lab is the investigation of the relationship between behavioral phenomenological patterns and brain functioning, as well as the investigation of the psychopathology and pathophysiology of psychiatric disorders, especially bipolar disorder. These two aspects are complementary, since the investigation of the neurobiology of psychiatric disorders may help to better understand brain functioning in general. And on the other side, studying the physiological relationship between brain functioning and behavior may provide relevant information on the pathophysiology of psychiatric disorders. We are conducting our research work mainly using neuroimaging and in particular magnetic resonance imaging. We mainly use resting state fMRI for investigating functional brain alterations in intrinsic brain activity and its organization in large-scale networks, and diffusion MRI for investigating structural Y-matter abnormalities. Moreover, we complemented these neuroimaging techniques with various laboratory tools for studying changes in neurotransmitter signaling. And finally, we also investigate immune dysfunctions and their relationship with brain alterations.
So, the starting point of our research is the psychopathology of psychiatric disorders, and in particular bipolar disorder. Bipolar disorder shows a very complex psychopathology, as it is characterized by distinct active phases of illness, mania and depression, that show very different and often opposite symptomatology, so that many patients can manifest hyperactivity, euphoric or dysphoric mood, and a thought pattern excessively focused on external environment, up to psychotic states with hallucinations and delusions of grandiosity or persecution. On the other hand, depressed patients usually manifest hypoactivity, anhedonia and apathy, and a thought pattern excessively focused on internal states and rumination, up to guilty and nihilistic delusions. Thus, our work is aimed at investigating the relationship between such psychopathological states and alterations of brain activity, in order to build a working model of the link between behavioral phenomenological patterns and brain functioning. Moreover, we are trying to integrate alterations in brain activity and neurotransmitter signaling with structural brain abnormalities and immune dysregulation in a coherent working model of the pathophysiology of bipolar disorder.
Hello, 안녕하세요. I'm Hyung-dong Park from South Korea. Welcome to my office. I investigate how the interaction between brain and body contributes our conscious experience. So as you can see here, our brain continuously receives signal from internal body. Like at each heartbeat, heart sends signal to the brain. And I investigate how such interaction between brain and internal body shapes or contributes our conscious perception and action. As you can see here, our brain continuously receives signal from our internal body organ, like a heartbeat or a lung. So for a long time, people thought the role of such body is very limited, like the homeostatic regulation, like controlling temperature and so on. But I investigated the possibility that maybe this such a brain-body interaction may be more important than previously thought. So for instance, I investigated the role of brain-body interaction in visual consciousness. So visual consciousness means like this vivid sensation we have when we see a flower. So I showed that this brain and the heart interaction shapes our visual consciousness. And then I also investigated self-consciousness, like the sense of our own body. So I reported that the interaction between our brain and our heart contributes the sensation of self-consciousness. And more recently, I investigate the role of brain-lung interaction in voluntary action. So I reported that the timing of our voluntary action and our respiration is synchronized. Okay, these are what my laboratory is doing. And maybe some of you wonder how we can investigate like self-consciousness. It's very complicated phenomenon. So for that, I often use like the advanced technology like virtual reality or robotics. So as you can see in this video, using virtual reality, we can manipulate this multisensory integration between vision and touch, which is known to be closely linked to ourself. So by using this virtual reality, we can experimentally investigate the self-consciousness. In addition, as you can see here, this robotic device, so using this robotic device, we can experimentally alter the sense of agency, the sense that I'm the subject. Okay, thank you. Bye-bye.
Hi everyone, my name is Christoph Dahl. I'm an associate professor here at GIMBC and I'm in charge of the lab Computational Ethology. I'm going to show you now in the next couple of minutes what we do, what kind of research questions we have, and what research models we use to answer these questions. The question is, what is computational ethology? To answer that question, we first need to address the question, what is ethology? Ethology is the study of behavior and social organization from a biological point of view, and therefore is part of the larger domain of biology. Now, computational ethology emerged out of ethology, and it's a relatively modern research domain. It is also an interdisciplinary research domain that addresses the lack of instrumental methods in ethology by introducing tools that originate in mathematics and computer sciences, in particular in artificial intelligence and machine learning. The defined goals of computational ethology are to detect predefined behaviors in freely moving animals and to discover novel behavioral patterns and new behaviors. To this end, it is essential for us to monitor the exact behavior and the spatial configuration of each individual in the group for a long period of time, at least several hours, during social interaction in both free behavior tasks or controlled experimental tasks. It is therefore essential also to use automatic tracking methods to elucidate the mechanisms of social interactions. We developed a real-time tracker that is capable of keeping track of individuals over a long period of time, and this is exactly shown here in the figures. Now, what we also do is we neurochemically induce pathologies in order to address potential impacts of neurological and psychiatric disorders on group dynamics and social interactions in these animals. In general, in my lab, we are interested in questions like social awareness, social decision making, group formation and dynamics. All of these questions are part of the larger domain of psychology. The only question that remains now is which animal species are going to be used in these studies. There are three main species: the jumping spider Phidippus rhesus, the zebrafish, and the Turkestan cockroach. In my lab, the goal in studying social cognition in animals is to characterize the smallest building elements of behavior and the behavioral control system that are made up on the basis of information processing mechanisms. A very interesting animal species when it comes to studying cognitively driven behavior in miniature brain models is the jumping spider. The jumping spider has an amazing approach to getting about. It jumps multiple times its own body length. On top of that, it has superb eyesight. It is extremely visual, almost unmatched in the invertebrate world. It also has a relatively large brain that collects information from the environment through multiple sensory systems. This in turn provides situatedness to the spider, and the spider is in full control of its environment. Now once again you can see how multidisciplinary this research program is. Computational ethology is rooted in biology. We apply techniques that are rooted in computer sciences. We add an additional dimension which is rooted in medicine, in translational medicine, by neurotoxicologically stimulate the animals. And last but not least, we ask questions which are deeply rooted in psychology. I hope you liked my presentation. And if you like any of these research projects which I proposed, or you have similar ideas along the same lines, then get in contact, or even better, why don't you go online and you apply for one of our programs at the master's or the PhD level, and then probably we'll see each other soon here in Taipei.